Alright, this will be the last segment of the Chapter 3 lecture. Moving on to nucleic acids. Nucleic acids are information storage molecules. They are what provide the directions for building actual proteins. This depiction of a certain nucleic acid should look familiar. Do you know what it is? This is actually a depiction of DNA. DNA has a double helix design to it. There are two types of nucleic acids rather. DNA, of course, stands for deoxyribonucleic acid, and RNA is simply ribonucleic acid. Nucleic acids are polymers of monomers called nucleotides. So I know that sentence might be kind of confusing, but you know that the word polymer means many units. Monomers are those building blocks that make up polymers. So the monomers that form a nucleic acid are nucleotides. A DNA nucleotide monomer consists of three parts. We have a phosphate group, a base, and a sugar. So this color coding is handy. Again, in this larger depiction, we have a phosphate group, we have a sugar, in this case it's deoxyribose, and a nitrogenous base, which is going to be adenine, guanine, cytosine, or thymine. In this case, it's thymine. Each DNA nucleotide has one of the following bases. Again, those are adenine, guanine, thymine, and cytosine. Nucleotide monomers are linked into long chains. The chains themselves are called polynucleotides, otherwise known as DNA strands. And a sugar-phosphate backbone joins them together. So you have alternating sugars and phosphates here, forming the backbone. The nitrogenous bases here. And a nucleotide, again, just consists of one of each of those components. So a phosphate group, a sugar, and a nitrogenous base. Two strands of DNA join together to form a double helix. And what's interesting about this is adenine always binds with thymine and cytosine always binds with guanine. So if you have one part of a DNA molecule, just a single strand, you can examine those nitrogenous bases and figure out the base that it would bond to. RNA is also a nucleic acid, but it uses a different sugar than DNA. Its sugar has an extra hydroxyl group, and it has the base uracil instead of thymine. So in RNA, you're not going to find any thymine. You're instead going to have uracil. So that substitutes for the thymine. Evolutionary relationships between organisms can be assessed by comparing their DNA. Through molecular genealogy, we can extrapolate relationships between various species, and biologists use molecular analysis of DNA and protein sequences in order to test their evolutionary hypotheses. So for example here, this phylogenetic tree, which is a depiction of various species and however many millions of years ago they branched away from a common ancestor to form their own evolutionary trajectory. And all of this information was determined by differences in the amino acid sequence. So if we know the amino acid sequence of hemoglobin in humans, we can compare that sequence to those that are found in different species and find out approximately when in our evolutionary past we diverged from a common ancestor. So for this case, we have rhesus monkeys here, diverged from a common ancestor around 26 million years ago. And our closer relatives, great apes, diverged about 8 million years ago, based on amino acid differences in hemoglobin. Alright, this is going to wrap up Chapter 3, Part 3. Students, if you have any questions or concerns or need more information about anything, please don't hesitate to email me, and you can also consult your e-text. Thanks for listening.